So topic 17, translation. This is going to involve the ribosome, of course. And this is kind of, like I said, one of the big three things that you need to learn and know really well for the final exam. Remember, we have replication, transcription, translation, okay? So make sure you know those concepts really well. Everything else in this last part of the course is all built around those three concepts. If you can understand what's going on with transcription and translation, then when we talk about viruses in topic 20, you're going to understand what the big deal is with viruses and so on. When we talk about biotechnology, it's making use of these kind of tools and these enzymes and those kind of things. All right, let us get into this and talk about translation. So there's our little diagram that I keep coming back to. And like I said, I'm emphasizing that these three processes, final exam, know them well. Going to be a big part of the marks for the final exam. So before we get into the unit, I thought it'd be useful to actually play the video first, one of these BioFlix videos. This one is pretty good, and it's going to introduce some concepts, and you're going to see some of the concepts that we've discussed already. So let me press play on that. Where is the play button? Usually it pops up here somewhere. Oh, there we go. All right, enjoy.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so there's a lot to unpack there. You can see a lot of concepts we've talked about already. Maybe you noticed the histone proteins at the beginning, and then there's the transcription, and you saw the splicing and all that going on. Today we're going to be talking about translation. So of course, translation takes place at the ribosome, and there's a bunch of components we need to talk about first to unpack this thing and understand what's going on. So we've got the messenger RNA, we've got the amino acyl tRNAs. So what does that mean? So this is just kind of a fancy way to say an amino acid is attached, right? The full word would be amino acid, but once it's attached, it's no longer an acid. So YL is kind of means a derivative. So maybe you're familiar with some organic chemistry. If you have a methane group, it becomes a methyl group, those kind of things. So what it's saying here is we have an amino acid attached to transfer RNAs. And then of course we have the ribosome and the two subunits. So let's talk about each of those. So first is the transfer RNA. So there's some things I want to point out on this. This is basically RNA, and it is has some single-stranded and some double-stranded kind of components. If you that first image is kind of a flattened two-dimensional structure. It kind of looks a little bit more like that structure in the middle, and because it's three-dimensional. And sort of that's the way it packs. The textbook, just to simplify things, kind of makes it look like that green blob on the right. So a couple things to point out. First is we're going to attach an amino acid here. It's actually the 3' end. And secondly, that there is an anticodon on the other end. So I can't remember how many transfer RNAs we have. We don't quite have as many as codons that we have, but we have more than 20. 20 amino acids, so we need at least 20 transfer RNAs. And this one here in particular, so that anticodon there, you can see it's AAG. And if I think about what the codon on the messenger RNA is going to be, it's going to be complementary to that and anti-parallel. So the codon itself will be UUC. So look this up in the genetic code, and that codes for the amino acid phenylalanine. So the cell will take this transfer RNA, and it's going to take phenylalanine, so that's what phenylalanine looks like. And there's an enzyme. You can see this enzyme here has that name in here. It says it's going to make an amino acid transfer RNA, and it's a synthetase. So it's an enzyme that's going to synthesize that thing, which is the amino acyl transfer RNA. So basically, you've got the transfer RNA, you've got the amino acid. This takes ATP, and actually this process will actually break the ATP down all the way to AMP, adenosine monophosphate. It actually uses 2 phosphates to break this whole thing down. And then you end up with your amino acid attached there. And if you're interested in chemistry, that's something called an ester bond. So this thing is ready to go. And like I said, there's a whole bunch of these enzymes doing this and getting these transfer RNA amino acid things ready to go for the translation. So they're all going to get shipped off to the ribosome once they're produced. So quick test yourself questions. So true or false? Let's take a look at each of these. It's talking about ribosomes. Ribosomes are made of ribosomal RNA and proteins. That is true. We talked about that already. Ribosomes are made in the nucleolus, or the nucleus. I guess I just gave away the answer. The answer is true. They're made in the nucleus, and they're made at the nucleolus. So I was just getting a little bit ahead of myself. Nucleolus, of course, being the component inside the nucleus where the ribosomes are made. Number 3, it says ribosomes can be cytosolic or bound to the smooth ER. That one's false. So what's wrong with that? Take a look right there. They're bound to the rough ER. The rough part of the rough ER is actually the ribosomes that we're observing when we look at it under electron microscope. So there's a ribosome. I think I showed you this image before. You can see it's made out of RNA and it's made out of proteins. And I want to just draw your attention to right there, and right in there somewhere you'll see this little green blob pops up once in a while, and that's actually the catalytic site, right? The active site where the new peptide bonds are going to be produced. So you may also remember that we have different ribosomes if you are a eukaryote or a prokaryote. The prokaryote ribosomes are actually a little bit smaller, and this is actually a drug target. So if you've ever taken erythromycin or tetracycline or a handful of other antibiotics, these antibiotics will interact with the ribosomal subunits, and they inhibit protein synthesis in the bacteria, and so they can kill the bacteria and clear an infection. So let's talk about the nucleolus and what is going on there. So that is inside the nucleus. And if you think about all the processes that are happening here, right? Proteins are made out in the cytoplasm somewhere, right? So that's where we have to start. We've got to make those proteins somewhere. But then we also have that RNA, and the RNA is going to be synthesized actually in the nucleus. So what happens is these proteins come in, they join the RNA, and then they make these little subunits. So eventually you get a large subunit and a small subunit. Then those are going to go outside a nuclear pore, and eventually they're going to get together and make a full ribosome. So they don't actually get together, by the way, until they have bound messenger RNA. And that's what we're going to talk about here shortly. So here is a model of a ribosome, and there's three things to notice, right? Three binding sites, and very conveniently they spell out the word ape. So that's always good when things spell a nice little mnemonic. So the A stands for amino acid, or technically it's amino acyl transfer RNA. So that's where the new transfer RNA carrying an amino acid is going to come. So like I said, think of A is where the new amino acid, A for amino acid, is going to come in there. Of course, it's attached to a transfer RNA because that's how this whole system works, right? P is for peptide. Okay, so you can see that the full name is peptidyl transfer RNA. It means there's going to be a peptide bond formed there. My mouth is just, my brain is getting ahead of my mouth, or my mouth is getting ahead of my brain today for some reason. And then E is exit. So if you remember amino acid, peptide bond, and exit, that's going to help you to figure out what is going on here. So let's take a look at the details. In terms of translation, I think in your notes I made kind of like a little bit of a flow chart in terms of what happens. If you read the textbook, they do give you more details. You're responsible for the details that are in the notes and in the lectures. So make sure you know those ones. If you want to know extra details, that's great. But it is going to talk about a few things like elongation factors that I am not going to discuss. So first step is initiation, right? So initiation just means we're going to get things going, we're going to start. So a handful of things come together during the initiation step. So you can see the first thing that is happening is we have the messenger RNA and we have a transfer RNA. And you can see right there, the codon, AUG, on the messenger RNA is going to line up with the anticodon on the transfer RNA. So AUG, and the anticodon is going to be 3' UAC. So exam tip. Last year, I think I had a true and false question, and I said, if the codon is this, then the anticodon is this. And so there's a good idea for an exam question. Not guaranteed I'll use it, but it's a good idea for an exam question. Okay, yeah, and so, and this lines up with the small subunit, and it lines everything up around the P site. The P site's in the middle, right? APE, APE. So it lines things up in the middle, and then it's kind of almost ready to go. We just need the large subunit. All this, of course, takes energy. So for whatever reason, we're not using ATP, we're using GTP. Like I said before, GTP is just like ATP. We're just using a different nitrogenous base, but in terms of energy, it's got 3 phosphates and 2 high energy phosphate bonds. So when we break that down, it's going to it's going to release energy, which can be used for all sorts of processes. So there it is, everything is ready to go. You can see you've got that transfer RNA in the P site, and we have this complex, and it has a name, translation initiation complex. People love naming things like that. It's kind of boring. I would probably call it something else, but I guess it's a good functional name. Okay, so we're ready to go. Now we just need more amino acids. So let's do this thing. Okay, so the A site, I told you before, the A site is where new amino acids are going to come. So there is a new amino acid right there. And This amino acid here is, it doesn't say what it is because we can't see the sequence, but what is going to happen is that there's going to be a codon here in the A site. So codon right there in the A site, and it's going to match that anticodon on the transfer RNA. So the transfer RNA, you know, I'd almost call it translation RNA because it's kind of like, this is the technology here. This is the technology that is helping us to go from one language to another, right? Because of that codon recognition step. So codon is recognized by the anticodon and the transfer RNA. So that's going to bring in the correct amino acid. So after that, you've got these two amino acids, and now they're, you know, like any good enzyme, they're bringing two things close together. By the way, every step along the way here takes energy. You can see GTP is being consumed here. We're going to consume some more unless it's ATP after this. And this consumes loads and loads of energy. So like I said, we've got the two substrates, they're together, two amino acids, and what we're going to do is this, you can see what's going on here, there's a little bond here, and this bond is going to jump off of this transfer RNA and go attached to this amino acid there. And so a new peptide bond is being formed. So that ester bond is being broken, peptide bond is being formed, and now if you think about what's going on, the little peptide chain is now attached to a transfer RNA in the A site. So at this point, we've accomplished in making the peptide chain one amino acid longer. So all we need to do is kind of reset and make sure things are kind of at the beginning of this cycle. So the last step is this, something called translocation, and that's where everything kind of shifts over. Right, so you can see my note there. It says the ribosome and the messenger RNA move in opposite directions. So it's kind of like, you know, somebody, you know, you're sitting on a bench and someone says slide over, and you slide over. And if you're at the end of the bench, you're going to fall off the end of the bench, right? So that's what happens here, is because we've got this transfer RNA right here, and it slides over to the E site, but the E site isn't really a binding pocket. Remember, the E site is the exit site. It's the door. It's the end of the bench. And there goes that transfer RNA. And so this is going to go, it's going to go and get recycled, and it's going to go back to one of those enzymes and get a new amino acid. And now, if you look, we have our peptide chain is going to be one amino acid longer, and that's good. And we're just going to repeat that because now we have another codon into the A site, and we're going to keep on going along, and as long as we have codons, we can keep adding amino acids. So some proteins are, you know, average protein is like 100 or 200 amino acids, right? Some are a little shorter. There are some in the thousands. It could be a lot longer. Just kind of depends on which protein it is and what the gene is saying, right? The gene could be, you know, like I said, could have many codons or just have a few. So this will go on again and again and again. You're going to elongate the protein. And then you probably know at the end of the gene is the stop codon. So what happens when we get to the stop codon? We've got three stop codons, right? We've got UAG, UAA, and UGA. And there's no transfer RNA that recognizes the stop codon, but there is a protein thing. It's actually called a factor because I think it has RNA in it as well. But it's called a release factor, and I believe it's mostly protein. This will actually recognize the stop codons. So this is kind of like, this is it. This is the end. This is the thing that stops the whole process. It basically puts the stop button on and tells everybody they're done. And so they just break apart and they go away. Again, takes a little bit of energy. And so your protein is done. It's going to go off and do whatever it does in the cell. Your transfer RNAs are going to get recycled. Your messenger RNA, all these things are going to go off and get recycled and reused for more translation. So that's the process of translation. I know it's a lot of steps, okay? And I know it's easy to get transcription and translation and all that confused, but it's important that you do go through and study these and know the steps and kind of the things involved. Like I said, it's going to be these replication, transcription, and translation are going to be a big chunk of the final exam. So it's important that you do know them. So I do have another video here. This one's a little bit shorter than the last one, maybe not as good resolution, but it's a nice short and sweet kind of 2-minute video that shows the process. And you know, maybe this guy explains it a little bit better than than I do, and I thought it was a pretty good video anyway, despite the fact that it's maybe not as sharp as the other one. So I'll play that for you right now.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
All right, so that's the end of that video. So there's a few more things to talk about translation. And I want to talk about, give you guys another problem to look at and talk about mutations. So one thing to note is that our cells like to be quite efficient. And this doesn't have to be done just one ribosome at a time. In fact, one ribosome can bind and another ribosome can bind shortly after it. And these have been observed in cells. They're called polyribosomes. In fact, you can see all three processes, initiation, elongation, termination, actually happening simultaneously, which I think is kind of cool. And if you are a bacterial cell, then why stop at that? There's no compartmentalization. So a bacterial cell can do transcription, so they can make the RNA. And before the RNA is even done, translation can start. So you can see that's happening here, right? We've got that little 5' end here. That is, this little 5' end has been produced. And, you know, as soon as the promoter and start codon are ready to go, a ribosome can jump right in there and start making proteins. So kind of cool, I thought. So this slide here is just kind of near the end of the chapter. It's kind of summarizing everything. And you can see all the things that we've been talking about. You've got transcription, you've got RNA processing, the introns being cut out. You've got capping and tailing. You've got a nuclear pore where it's exiting, and you've got translation all there in one step. So you can take a look at that. We'll review the videos. Like I said, I want to spend a little bit of time today talking about mutations and a few other things, of course. So here's our protein, and you can see it's folding up. And just a reminder that there's kind of two scenarios here, and we're going to talk about these scenarios here next lecture, where proteins are sometimes just made, and that usually involves cytoplasmic proteins because they're made and they're ready to go. But many proteins are processed. So we talked a little bit about glycosylation. That's where we're adding carbohydrate And we talked a little bit about how some proteins are sent off to certain parts of the cell. And like I said, we're going to talk about that in topic 18 in a little bit more detail in terms of how that is done. So more on that next week. So like I said, I want to kind of, spend the rest of today talking about mutations. And so let's take a look at the consequences of at least one mutation here, and then I want to talk about some different types of mutations. So one type of mutation, sometimes mutations can be quite detrimental to a cell. So let's take a look at this particular gene. This is the beta globulin gene that's part of hemoglobin. And we're looking at one amino acid in particular. So if you take a look, we've got, there's the DNA right here, and the RNA is coded as GAG. You look that up on your genetic code, and you're getting this amino acid here, glutamic acid. So that's a charged amino acid. That's kind of an important detail. And so let's look at what happens if you have sickle cell anemia. So sickle cell anemia is a recessively inherited genetic disorder. So they have one base pair change. So right here in the middle of that particular codon. So instead of a GAG, we get a GUG. And instead of a glutamic acid, in the end, we get something called a valine. So what? One amino acid. How does that cause these kind of funny looking cells? The disease is called sickle cell anemia. So just before I get any further, let me tell you what a sickle is. So a sickle is kind of like a tool, kind of looks like this, right? It would be something that maybe you see the Grim Reaper would have, right? It's got a big blade on it for cutting, wheat or grains by farmers. And anemia means you don't have enough blood, or at least blood cells. So let's talk about that, how this works. So this is called a missense mutation, by the way. I'll get back to what that means shortly. So what is going on with this sickle cell anemia? So I told you glutamic acid is a polar charged amino acid. Valine is hydrophobic. So this is bad because you're going from, you're changing the chemistry of this thing, right? You're going from something that is polar charged and water soluble to something that is hydrophobic and not water soluble. So it turns out that valine is kind of, you can kind of think of it as like a little sticky patch, right? So it's sticky and it will stick to other valines and it will avoid water. And that's exactly what happens. So you can see here, it's the hemoglobin molecules and they're kind of clumping up, right? Hemoglobin should be soluble. It's trying to grab your oxygen and bring it from the lungs to your tissues. In this case, it's clumping up and forming kind of like these, well, we call them fibers, right? Because they're forming long strands. And you can imagine what that does to a cell, right? So here's your normal cell right here, and you've got soluble proteins that are floating around, and the cell is kind of a nice shape, and it fits through your capillaries very well. The sickle cell has all these fibers in there, and they're kind of forcing it into these funny shapes, a lot of them that look like sickles. And these cells become a lot more fragile, and they break open, and so the person can become anemic. Apparently, it can be quite painful at times, and of course, the person's health is suffering quite significantly. So this is something, by the way, that's you see this in a lot of biology textbooks because it's a very well understood genetically inherited disease, and it was actually the first one ever characterized at a molecular level. So yeah, we don't want to have sickle cell anemia, or probably we don't want to have it. But there's something else kind of interesting in terms of genetics around this thing. I just wanted to mention that, so you may wonder who is likely to get sickle cell anemia. So if you take a look at that that map on the left, it turns out that if you're from some of those geographical regions, particularly sub-Saharan Africa, there's a high prevalence of sickle cell anemia. And it turns out that if you look at the geographical distribution of malaria, or at least historical distribution of malaria, it kind of fits the same map. And so we've learned in the past few decades that people who are a carrier of the gene they seem to be a lot more resistant to malaria than people who don't have a carrier of that gene. So it's probably one reason why this particular gene has persisted in the human population, particularly of people of this kind of geographical origin. So kind of interesting anyway, but like I said, we probably still don't want it. It's not a good disease to have. So as I mentioned, this particular kind of mutation is called a missense mutation. So I want to kind of just talk about these different kind of types of mutations and scenarios, and then, like I said, give you a problem. So you can see missense is the second one here that we're going to talk about. But let's take a look at these scenarios. So first scenario of mutation is something called the silent mutation. So there's a gene we're going to look at. It doesn't really matter what's coded on that gene at the moment, but you can see we've got, it's very, very short. We've got a start codon over here, codes for methionine. We've got lysine, phenylalanine, glycine, and then eventually a stop codon. So take a look at that scenario, and let's make a mutation right here. So we're going to change this GC base pair to an AT base pair, right? And so if I take a look on my genetic code at GGC, so GGC is this one here, and then I look on the genetic code at GGU, turns out they both code for glycine. It's the same amino acid. So this is a silent mutation, meaning it doesn't really matter. The glycine is a glycine. The protein is getting the same amino acid. There's no biological effect. So this is a silent mutation. This is the best one to have. There are thousands and thousands of these characterized out there in the Human Genome Project, because of course it has no effect, and therefore it can be easily occur and transmitted from parent to child. So we already talked about the missense mutation, and in this case here you can see what we're doing is we're changing this base pair, the CG, to a TA, and we're getting glycine to a serine. So that was the same type of example with the sickle cell anemia, and this is called a missense mutation. So missense, I gotta love these funny geneticist words. I really don't like the geneticist words, but missense, right? That's just kind of the first half of the word mistake, right? A mistake was made, wrong amino acid, okay? So hopefully that helps you to remember which one this is. There's also a nonsense mutation. So nonsense starts off with the word no or none, okay? So what's gonna happen here? So let's take a look. So what if we make this change? So we're gonna change this TA to an AT. So now our codon is AAG, and I'm changing this codon to a UAG. So hopefully remember your genetic code. The UAG codes for stop. So none, right? No amino acids are made after this point. So this can be really bad. Sometimes it's not so bad. I mean, if you're only trimming off like a couple amino acids at the end, the protein may actually be functional. But in many cases, the protein is actually truncated, which means it's cut down, and it's totally non-functional and leads to sometimes some severe biological effects. So I'll show you one example of a mutation that involves a nonsense mutation. This is a genetically inherited disease, a certain type of muscular dystrophy. And this is an X-linked form of the disease, so females don't usually get it because they usually have the one good form of the gene. So sometimes you see this in males, and these people have severe developmental issues with their muscles and all that. So I want to talk a little bit about one other kind of scenario is sometimes you get insertions or deletions in mutations. And so you're getting some of the same effects. And let me just show you how that works. So here it says a frameshift mutation. So take a look at what's going on here. I'm just taking this same sequence and I'm throwing in an extra A there. So if you think about this, AUG is all good. Everything else is all messed up. In fact, in this case here, the very next codon, I've turned it into a stop codon. So obviously that's going to be bad. These kind of things happen once in a while. Like I said, usually you're looking at the entire gene as non-functional. It can be lethal to the cell. Sometimes we end up with extensive missense. So you can see in this case here, we've got a whole bunch of wrong amino acids. Once, in this case here, what they've done is deleted this A here, right? And after that, all the amino acids are wrong. So you got the wrong kind of protein going on there. Once in a while, whole triplet is either deleted or added. And so you can see in this case here, this triplet here, TTC is missing. And you're missing amino acid. And again, this is kind of the scenario where most of the protein is good. But if it's a crucial amino acid, we could be in trouble. Okay, so I'll show you an example of this, again, in a genetically inherited disease. This is called cystic fibrosis. So this is the most common recessive genetic disorder amongst Caucasians. So about 1 in 700 people are carriers. And if you have 2 copies of the gene, you end up with cystic fibrosis. So this affects a chloride channel. And so all your secretions are messed up. And so digestive secretions in the lungs, and a lot of these people end up getting really thick mucus in the lungs. And the lifespan is about 32 or something like that. And often what gets them is an infection in the lung that cannot be treated. Okay, so different types of mutations. And my plan was to show you another problem here. And we're gonna take this up next day. I see that an administrative assistant is trying to get a hold of me because she wants to do the class evaluation. So I'm gonna let her into the meeting. But before I let her in, I'll just say I'll come back to this question next day. And you've taken a look. I've also put an exam study guide on Moodle. And it has one of these translation, transcription type of questions on there that you can take a look at and with an answer scheme. One more note before I let Ruth in is I sent out the information about the lab exam yesterday. So hopefully everyone got it. Hopefully you can read all the instructions on it, and maybe I'll give you some more tips next week. But take a look at your previous lab reports and all that, and take a look at the email for the instructions. So I'm going to let Ruth in now, and like I said, I think she's doing the class evaluation.